All right. We're going to talk a little bit about bioinformatics today. And bioinformatics is, in general, the concept of combining biology and computers together. It is a rather complex set of, computers, supercomputers, databases that all work together to do anything biology related. And just as a quick reminder, Skynet is real. Anyone who says otherwise is wrong or getting hacked by Skynet actively. Okay, so that aside, so what is bioinformatics? Essentially, you can chalk it up to the concept that a group of biology nerds thought to themselves, hey, could we be any nerdier? And one of them suggested adding computers to what they were already doing. And now you have ultra nerds, or bioinformatics. And realistically, that's actually kind of what happened. They were basically like, hey, we're generating a lot of this data. This data is getting more and more difficult to handle. So they went about and said, hey, we're going to analyze these very complex data sets using computers because it's eventually going to get to the point where it's beyond our ability to handle. And at the time, the very early age of bioinformatics, this was like, well, you can actually just do it by hand or do it by small amounts of computer interaction. You don't need a lot of computing power. But now it's, definitely gotten to the point where we absolutely have to rely on these huge databases and searching abilities and kind of developing, even to the point where we're getting to AI-assisted analysis of these things because it's so completely difficult. So we are currently sitting in the golden age of bioinformatics. We've made such huge discoveries and huge strides within bioinformatics that it's unparalleled. We would not be where we are today in our understanding of biology without bioinformatics as a key component of it. It goes everything from genetic analysis to modern protein synthesis. It goes from large population genetics and predictive analysis of different diseases and epidemiology. It's massively important now. So we're going to talk a little bit about it and kind of the fundamentals, kind of the easy introductory stuff, which is still kind of complex, but just bear with me as you go through it. So who runs the bioinformatics for the country? Well, one of the big things is NCBI. NCBI is National Science for Bioinformatics, or National Center for Bioinformatics, is funded and run by the National Library of Medicine, which is funded and run by the National Institute of Health. Now, the total budget for the National Library of Medicine is $380 million a year. But the NCBI is only a small sliver of the NLM, so they even get less. So we don't actually know how much they get. But we can say it's definitely less than 380 million. I believe this is the 2020 figure. It may be a little bit more, but more or less it's about 300 to 400 million. So that becomes really, really complicated. How did it get started? Well, Senator Claude Pepper was like, hey, we should really start investing in this early. And he created a bill to create NCBI as part of the National Library of Medicine in 1988. And yes, these do get really, really complicated. All the biggest things that you have to memorize is National Institute of Health and National Center for Bioinformatics, or Bioinformation rather, Biotechnology Information. The general mission is to create new information related to nature. That is it. So it's a very broad, it gives them a lot of room to move around. So that's great. The most important and common feature is BLAST. BLAST is a massive database search engine. So it's an algorithm that searches a database and hosting pretty much all genomic data created anywhere in the world that submits it. So basically anybody in the world can go to this database and go, hey, I would like to submit the sequence that I've made. I've followed the proper protocols, here's the admission, and then they will host it. So that means that anybody else in the world can search and find that, which is very, very useful because searching tons and tons of information from all over the world for free is really key, important part of it. And without free and universally available resources, we would not have sequenced the human genome as fast as we did. The reason why we were able to do this is because we were able to generate primers and new stuff, kind of collect all the data from all over the world and generate a whole genome setup from the human genome because of this. And furthermore, it's also helped us sequence so many other genomes faster. It has been a crucial part in understanding how all of these genomes work and getting more and more information and figuring out what is a gene, what is an encoding sequence, what does all the stuff in between genes mean, and having this massive database that's for free for anybody in the world is actually a really huge part of this. They're not the only database in the world, but they are one of the largest databases for just genetic information, period. There's some other ones like Japan does a lot of enzyme database stuff. I believe Europe, EMBL has their own protein kind of focused one, which has to do more with 3D structures, but there's a lot of different sharing throughout the world, and this is kind of the big one right now for DNA specifically. So what is BLAST? BLAST stands for Basic Local Alignment Sequence Tool, or nerd matching for short. This is important to remember, so remember this acronym, Basic Local Alignment Sequence Tool. What does it mean? It's an algorithm that bases pairwise alignment of sequences, so it basically looks through a bunch of different sequences and says, hey, are these close? And then it gives a value on how close it is, and then it kind of goes down the line of that sequence and gives it a value of how close it is to another one. You can do this for RNA, you can do this for DNA, you can do this for protein, you can do this from protein to DNA or from DNA to protein. And this becomes really important because, of course, you know that DNA, will have a direct translation to protein, but a protein, because there's different sequences that can lead to the same protein, going backwards is a little bit tricky, but we can do that as well. There's a bunch of different formats that are accepted. The big ones, though, are FASTA and GenBank, and I'm just going to talk about those two. The way the format looks, FASTA is just basically the easiest one. It's just a little greater than sign, and then a title, and then a new line, and then the sequence just straight in a row. This is actually the easiest one. That's like basically the only aspects to it. It's actually really, really simple in that sense. Now, the other one is GenBank. GenBank has a bunch of other information sets within it, so it'll have different details. So like here it talks about the exon level things like, hey, look, here's a region within this gene for SOD. It's a homodimer. It's part of HSOD1. Here's another exon within it. So it's saying, hey, look, the sequence jumps from here to here in the actual genome, but this is the actual coding sequence. And then it'll have the origin sequence, what the gene looks like by itself. So it's basically the copy of this thing. The difference here is that they do this by 10. So for the GenBank format, it's like, here's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So I know right here is where we start with 10. So 1 through 10. So it starts right here, right? And so this way I can count really quickly. So I know that right here is 61. That means over here is 60. So if I was trying to look for the sequence that starts at 238, I can go, okay, well, over here is 241. So that means that this is 240, this is 239, and this means it's 238. So it's really convenient for finding that. So it's easier to read for humans, but it's not really as useful for machines. So it's got a lot of information to it, but it's more human friendly. This is more machine friendly. They don't really care about all this spacing. They rather not have this at all. So you can see that they're all pretty similar, but one's more readable for humans and the other is more readable for machines. And this one's a lot easier to type out. So most of the stuff that you guys will be putting into BLAST will be just a little greater than sign and then a title. The title can be as long as you want it. You can put literally any characters in that you want. But as soon as you hit new line, then it has to be sequence data. And in this case, the sequence data is DNA. Okay, so this is what the BLAST website looks like. And I'll kind of give you guys a walkthrough of this too. But you can see that there is a ton of options here. It is lots of different things that you can change. It is very confusing. And this makes it daunting for a lot of people. We will go over a simplified way of this. And there are videos on Lab Archive that show you guys how to use this because this is very, very confusing. So I highly recommend you watch through those videos, but I'll give you guys a brief overview in a little bit here on how to use this. And there are significant settings that you can tweak, and we won't even cover all of those. Now, how do we get these sequences? Well, the way you do this is through PCR. And there's kind of two basic steps with PCR. There is just creating a copy, and then there's the sequencing aspect of PCR. Sequencing aspect is how you get the actual genetic data, but a lot of times when you sequence, you have to do the PCR first to get a small fragment that you want to look at, so that you get a nice, even section that's consistent so that you can do the sequencing and the sequencing isn't interfered by anything else. So we want to get a fragment. The way we do this is we basically run through a series of cycles, which we'll do later in the semester and I'll talk more about in depth. But basically we split apart DNA, we put these little moon things here, which are DNA polymerase, and they basically make copies of a region. And they start with this primer bit, and then they put in nucleotides that go in the right spot, and you make a big long line. And eventually it falls off, and then you do this copy again. And you do repeated cycles, and the more cycles that you do, the more copies of this little region that you get. And so eventually you'll just get mostly just this little tiny region so that you can look at it on a gel. Now, and that's basically the definition of this. So make sure you guys remember what PCR stands for: polymerase chain reaction, synthesis of DNA sequence from a template to create multiple copies. The more complicated aspect of this is that we can do different types of PCR. So we can use RNA as our base, or we can do DNA as our base. We can use double-stranded DNA, we can use single-stranded DNA. There's a lot of different things. It gets more complicated, but basically that's the easy kind of information. The end result that we get, though, is going to be DNA. So all of our PCR is DNA focused. We can analyze the copies made in order to gain more information about the source or the template DNA. So there's a lot of different things we can do here, which we won't go into too much detail, but we will kind of give you guys an idea. Now there is an electronic version of this. Because we have so much data built up, we can actually predict what will happen in a simulated polymerase chain reaction. So this is actually really, really helpful. We call this in silico. So anything we do in computer is called in silico. You know, there's of course in situ, which is in a living organism, in vivo, in vitro. Those are all different types of experiments. This one's a simulated in computer. So silico, silicon, PCR amplification. Now this website's really interesting because it has a whole bunch of genuses in it. We can go look through them and choose them for a specific organism, and then we can test a primer that we create to see what kind of bands we can get off of here. And there's a lot of things that we can set in here. So we can set what our target primer sequences are. So we can code those in here. We can then set our organism, so we can set it species specific within the genus. So there might be Bacillus anthracis or Bacillus subtilis. And then we can say, hey, also include plasmids, because plasmids are kind of separate from the genome, or we can exclude them because sometimes we don't want the plasmids. It just depends. And then we can say, hey, allow some mismatches. So the reality here that we were talking about before with this PCR, it lines up really nicely and matches perfectly. But the reality is, in nature, the primers don't have to be exactly perfect. They just have to mostly match up. And we can allow some wiggle room in here depending on the length of the primers and what have you. So we can actually set some. So we can say, hey, you know, if it has a mismatch, that's okay. As long as it's on this end of the primer, it's fine. Or how far from the thing. So we can set the stringency. Basically, how close do we want this? We can have it match perfectly, or we can have it match mostly. And part of the reason why we want to do this is each bacteria might have developed a couple of mutations somewhere within the sequence that we're looking at. So we want to make it, basically kind of give it the best option of what the real world reality might be. So we can say, yes, we can make this really, really stringent, or we can make it really, really loose and just kind of give us a better idea of what we might get. And we can say, hey, what's the maximum length of bands? Because sometimes you'll get bands that are huge, and because this is in silico, that means the computer's just predicting. So the reality is that we have kind of a limit of how large of a band we can create with PCR. So we can get some very, very large bands, but there's kind of like an upper limit. And sometimes we really only care about the small bands because that's what we're going to be actually using for our experiment. So just keep that in mind. So generally we set it about 2,000, 3,000 nucleotides. And then we can say, hey, let's go run this. So we hit amplify. And what it's going to do is it's going to produce, if the PCR is successful, an example of what you'll see in the gel. So when we actually run a gel in real life, this is kind of what you'll see. So here we have ladders on either side that tell us kind of what size bands we have here. And then here we have test things. So you can see this band is larger than these bands. And as it runs through the gel, the larger bands lag behind because they don't move as fast, and the smaller bands move faster. So we can see this band right here is slower than these two bands on either side of it. These two bands are about the same size. They will give us a gel image that's actually quite a bit like this. Now, it'll also tell us, hey, here's the length we'll see. Now, this is the really key part. It tells us the position that we will find this in. And you can see that there's actually a bunch of positions here, but they're all the same size because a lot of these will be copies of the same gene. We'll have multiple copies in the same genome because sometimes that happens if it's just by chance it has a copy of that one. Sometimes that gene's so important that we need to have multiple copies so that even if one of these copies gets damaged, we'll still have a functioning gene in there. And so you can see in this particular instance, right, we have a bunch of amplicons on here, and you can even click on here to see the position in the sequence. And here you can see where all the bands are. And so these are all overlapping bands, and they're all about 1500, so you can see them right next to the DNA ladder. It's at the 1500 band. And so we can see that this gene is 1500. Now, if these were a little bit more separated out, you might be able to see more bands in here, but because they're all basically the same, very tiny differences, you won't see those different bands in there because that's too small for us to resolve easily on a gel. So we'll have our simulated gel, we'll have our sequence, we can actually click on that sequence and we will get a little pop-up of what it is. Some other things that are kind of like this, that utilize this. Why is this still important? Why are just finding these bands important? Well, it's still one of our go-to ways to track diseases. One of the ways we do this is with this thing called PulseNet. And we do have a sequence database for diseases submitted to the CDC, but we also have these pulse field gel electrophoresis. This is the old faithful way, and it gives us a fingerprint of the DNA that's very, very, very specific to individual organisms. Eventually, we'll move to the point where we phase it out and we just rely on sequencing, so targeted sequences or whole genome sequencing, because we're just getting faster and faster at it and gives us more information. But we still currently work with pulse field gel electrophoresis, which basically says, hey, we're going to make a couple of copies of their genome and separate them out, and we can figure actually out subspecies information. So we can actually detect very, very minute changes within this. So even if the organisms are the same species, they might have like one subspecies that's this way and another subspecies that's a different way. We can actually tell that faint difference based off of those organisms. And the way it does it is with a gel that's very similar to the gel that we will actually run in lab eventually. Basically, it has a negative side and a positive side, and it pulls the DNA down. And where we usually do it is we usually do it on the top end, we put the negative, and the bottom end we do the positive. It's just one straight line. But here we can kind of shift it back and forth, and we do pulses. So we pulse it back and forth at 120 degrees, and this pulsing makes the separation a lot more clear and gives us much thinner bands and better resolution. And that way we can figure out basically the difference. This image is stolen from this YouTube website, so I highly recommend you guys watch this channel because they explain it in a little bit more depth. But basically, that's what you're doing here. You're just creating pulses to separate it out, and then we submit these images to PulseNet, and that way we can track diseases throughout the country. This is run by the CDC, and so by submitting this, we can find out if there's like an outbreak of, say, salmonella. And because we can get a very close fingerprint of this, we can go, oh, this is the fingerprint for salmonella, and they're all related in this one region. Oh, so these are all connected to the same kind of outbreak. So that's why this information becomes very important. Okay, so our cool science thing of the week. This also has to do with BLAST. I'm going to actually show you a real-world example of using BLAST. So these guys came out with an article. They said, hey, we found this gene. So MSH3, which stands for human mutagen S homolog 3, has a lot of homology and potential recombination link to SARS-CoV-2 furin cleavage site. So in SARS-CoV-2, we have a whole bunch of genes in there, and there's one gene that's for this important part called the furin cleavage site, which is really, really crucial for infection into humans. Without the proper sequence in there, it will not work properly or as well for humans. So it needs to have that cleavage site there to work. What these guys did is they were like, hey, let's go through SARS-CoV-2 genome because we have a whole bunch of these genomes and kind of compare it. And they said, okay, well, we know it's got about 82.3% amino acid identity to bat coronavirus SL-CoV-Z-C45. So it's got a lot of protein similarity to this normal bat coronavirus. So basically what we can say, this is kind of the canonical bat coronavirus. If we look at it with the amino acid identity to SARS-CoV, the original SARS virus, it's got about 72%. So it's got a significant amount of change. And we notice that it's very, very, very close genome sequence. So that's a step above amino acid sequence, because amino acids can have multiple different sequences that lead to the same amino acid. So this genome sequence identity is more stringent. So it's 96.2% identical to bat coronavirus RATG13, which is very, very close. But there's still some big, it may not seem like 3.8% is a lot, but it is quite a bit, especially if it has to do with specific genes. So while there is numerous point mutations differences between SARS-CoV-2 and this other bat coronavirus that we found, that we know exists, there was only one insertion and dissimilarity that exceeded 3 nucleotides. It was a 12 nucleotide sequence that was coding for 4 amino acids. So that's actually really important, that it's coding for amino acids. So it's not just something that's in the sea of genes, because most of the DNA is non-coding, although for viruses it gets a little weird when we talk about non-coding regions, because sometimes they're really important even though they're non-coding. We'll get to that later, another day. So it's coding for 4 amino acids, which is PRRA. And we know that PRRA is really, really close, encodes a 100% amino acid match for human mut-S homolog 3. This is involved in DNA mismatch repair protein, part of this complex. And their thing is, hey, we looked at this and we found that this sequence, so this nucleotide sequence, which is the same one up here, there's an absence of that in any other eukaryotic or viral genome in BLAST database that makes recombination in internal intermediate hosts an unlikely explanation for this presence. And so what they said furthermore is, we said, hey, we looked at the sequence identity. We said, looking at it, we saw that it extends beyond that 12 and goes to a 19 nucleotide sequence. And we did some math, and we did a bunch of math, and boy, it seems really, really unlikely that this happens by chance. So the assumption is, hey, somebody might have put in this nucleotide sequence. And so they did a bunch of math. Now, what this genome part is for, so this is the part that they're talking about, this cleavage site, it's really, really crucial for infection with COVID. So that's why they're bringing it up. And they're kind of hinting that, hey, maybe somebody was messing around with the genome. Now, why is this important? Well, because they have some problems here. First, the big problem is their math. They're like, hey, let's look at the likelihood of 19 nucleotides in a row in a 30,000 nucleotide sequence for the genome. And that's about how big the genome is of COVID. And they said it's a 1 to 10 to the negative 7th likelihood. Okay. Let's look at our next parameter. 19,000 nucleotide sequence appears in a 3,300 nucleotide sequence. That's the sequence for a gene, in this case, and that's 1 in 1 to the 9 times 10 to the, I believe, 8th. So that's very unlikely, even less unlikely. Okay, well, let's look at this and see how likely it is that this sequence would appear in one of the 24,000 sequences of this gene. And it's not, it's better, so it's more likely to happen. How likely is it to appear in 30,000 sequences of a genome in a library of 24,000 sequences with approximately 3,300 nucleotides? And it's very, very low. So their thing is like, the likelihood of this happening is very, very low. But the problem with this is that they're just using raw numbers. And the problem with using raw numbers, especially when we're talking about proteins, is because proteins are not nucleotides. So nucleotides can all kind of ending up making, they can have different sequences, but end up making the same amino acids because there's, some overlapping sequences that make the same amino acid. But on top of this, nature is really good at cheating and using the same sequence of protein over and over and over again. If you look at your immune system, the sequences you will find are almost identical for a whole bunch of immunological receptors, because nature is lazy and it will just use the same thing. So finding a protein sequence that matches, it's just it just happens. It's not crazy. So their math doesn't work out, even though it's technically correct, it doesn't technically reflect the reality of the world. But hey, maybe they're 100% right. So what do? Well, they gave us the sequence. So let's go look at the sequence here. So what we're going to do is we're going to copy this sequence. So we're going to say, all right, let's go and grab this sequence here. So we're going to copy their sequence. So this is just kind of the reverse of that, in case you were wondering. Or we can even just search with this one. So whichever one you want to do, grab this one. Okay, and we'll head over to BLAST. So here's BLAST. You can just search for BLAST and find it, no problem. You can say, we're going to do a nucleotide to nucleotide BLAST. And we're going to say, all right, we've got test sequence. So we'll make our title. So this is the FASTA format. So greater than sign, title, new line, and we're just going to cut and paste. So it's a really simple one. Now remember, one of the things that they said here is the absence of this sequence from eukaryotic or viral genomes in the BLAST database makes recombination in an intermediate host an unlikely explanation. They're basically saying this shouldn't ever happen in nature because this sequence is really, really unique. Okay, it's really, really unique. Well, let's go check that out. So we're going to say, hey, and you can see I've set this up before. We've got our non-redundant database. So this is the database that's just single copies of genes. There's no extra stuff in there. And you can make different ones based on what you want to look for. But this one, we're going to go non-redundant. We're going to say we're only going to look in eukaryotes. And we're going to just exclude humans because they said, hey, this has a lot of homology with the human one. So we're just going to cut out humans from this. So we're not looking at any humans. So we're only looking at eukaryotic organisms. So we selected eukaryotes, and you can actually just add more things in here if you really wanted to. So we could say, hey, add in viruses or whatever. And you can just search for the word and it'll let you find that. So if I just deleted this and I typed in eukaryotes, you can see that it lets me search everything in eukaryotes or eukarya. There you go. And we can pick any extra things. We can look for highly dissimilar sequences or more dissimilar ones. We're just going to do highly similar. We're going to hit BLAST. And so we submit this. Now remember that this is kind of a free search engine, right? Looking through a huge database with a very complicated algorithm. We have to wait in line. So we're just going to sit here and chill. It should take only about 30 seconds. And while it's going, we'll talk more about this. Whenever we put a publication in Science, we always want to verify stuff. And so this article I chose because they were kind of suggesting, hey, maybe somebody was messing around with the genome and then it started infecting people. So maybe this has that. And that could be it, but they have some big gaps. So we talked about the statistical gaps. You can't just do statistics on nucleotides because that doesn't make much sense, especially when we're talking about large streaks of amino acids and also, similarities in proteins because certain proteins kind of get copied a bunch. There's certain regions in proteins that get copied often. So that kind of math it's not very good. The other thing that we're going to look at here is, how accurate their statement was, which is it's absent in other eukaryotes. So I'm going to give this a little bit more time. I did a couple of searches before this, so they might be pushing me back. Oh, there we go. So this is what the results page looked like. It says, hey, here's our job title. Our job title is sequence. It'll actually give us a link so we can always go back and look at this for the rest of time. It tells us what we use, which is BLASTN. We're looking only through the nucleotide database. And this is our query ID, so we can actually go back and look up this query anytime we want. And we can look at the matches. So here's a whole bunch. It gave us kind of the first 100. And it tells us a bunch of values. It gives us our E-value, which is how confident we are. And then percent identity, which is actually the really easy way. So you can see that there is a whole bunch of 100% identities in here. And going all the way down, it's not a lot. But also it's going to look at query coverage. It's basically saying, hey, how far does this cover? So percent identity tells us, hey, this was a perfect match for all the sequences we look at, but then it's like, hey, how far into this query did we get to get the perfect match? And so in this case, the first few hits are actually 100%. And so there's a whole bunch of different things here that we can look at. So we can look at this one. So Drosophila fly. So we can look in here and it'll give us a picture of what that analysis looks like and how many identities there were. So it was 18, 100%. We can look up here and at the different organisms, we can actually look at the gene if we really wanted to. But that gene bank data, so you can see that's the gene bank format, is just a little hard to read, but it's a little bit easier. Here's the people that published that sequence, what have you. This guy is a fish parasite. I think it's like the white fuzz that grows on fish sometimes. I believe this is bird. So this comes from bird. And so you can see actually a whole bunch of different ones. If we look farther down here, this is a different type of parasite. If we scroll even farther down, Bos taurus, so that's cow. Even further down, this guy's elk. Bobalis is, I believe, water buffalo. So you can see that right from the start here, the fact that the sequence doesn't exist anywhere else, that's not really true. It's false. It does exist. And so we can see that. We can also see a graphical summary that shows us the alignments. So you can see that it kind of fits along this query quite well in a lot of these things. It hits right in the middle. You can see the other alignments here. You can see the taxonomy, and it kind of tells you the breakdown of what this looks like. You can look at organismal taxology and then the taxonomy here. Like you can see large groupings of the number of hits within each of these taxon. There's a lot of information here. But basically, this is all you have to do to kind of get a quick and easy way of saying, okay, well, here's 100% identity, here's 100% coverage, which organism it comes from, where the sequence is, what have you. And so it's actually pretty evident that this is not a unique sequence. And so what we've actually found out was that these guys retracted their paper. There was a whole bunch of problems in here. If you look at the authors, only the first author was a biologist and everybody else was some other non-biology related thing. So there was like a mathematician and a physicist, and it just didn't make a lot of sense. This was not a good article. So number one thing, whenever somebody publishes something in science, go back and look at it, be really careful, and then double check their work, especially if it sounds kind of fishy. Like that sentence, which I just kind of copied out of here, the absence of this sequence from eukaryotes and viral genomes, that made me go, that doesn't sound right. So there you go. All right. Now, the very last little bit before I let you guys go is what we'll be doing on the second day. So for 4102, you guys only meet on Wednesday. You guys will only be doing the lab assignments. So you guys can bring your computers in to work on this, all the bioinformatics lab steps on that one day. But the first thing that you'll do before you start all of that is that you guys are going to make copies of your slants, so we can have fresh, actively growing copies of your slants so we can do your unknowns. So you're going to make another copy of your unknowns, and you're going to grow them in the incubator so we can use them for the week after. 1102, you guys will be doing one part of this lab on Tuesday and the second part on Thursday. On Thursday, we'll have you guys make a copy of your slants and grow them in the incubator as well. If you guys have any questions on this, and I know it's really confusing, definitely make sure you ask your TAs. Come shoot me an email or something, and we'll go over this. Other than that, have a great and safe weekend for Easter, and I will see you guys next week.